Right, this is video 707, changes in the genetic code, and we're going to talk about changes at two different levels. And the first is at the chromosomal level. So we're talking about changes in chromosomes. And what I have here are four karyotypes that represent chromosome disorders. So we know that there are 23 pairs, and each pair should have two. So here's pair one, pair two, pair three, so on and so forth, pair four, pair five, for a total of 46 chromosomes. So what can happen is, we talked about during meiosis, during the first division, we have our four copies, and they line up in a tetrad, and we get crossing over. And two of the copies go down to for the second division, and then after that division, we go to here, and these get pulled apart then again, and we end up with four haploid cells. So, oops, I'm off screen a little bit. Let me just change this. Sorry about that. What could happen instead of two of the chromosomes going one direction and the other is you can get an error in a split, and you'll get three chromosomes going one way and one chromosome going another. And what that can result in is having three chromosomes or one chromosome. So the end result will be a gamete, or after fertilization, I should say, you'll have a zygote that has either three or one. So when you have this process is called non-disjunction. And really, all you guys need to know for your test is that it's a mistake during meiosis when the chromosomes separate. So it's an error in chromosome separation. And I have listed here four different conditions that we can have. So if the non-disjunction occurs at the 21st chromosome, and you get three copies instead of two, you have Down syndrome. And most of us have seen someone that has Down syndrome. We know that it's a person that has like eye folds, they have short hands, and there's often mental retardation, and that the brain and the abilities don't develop as it would in somebody that has two copies of the chromosomes. Over here to the other three, here is, we have a single X chromosome. So these are all on the sex chromosomes. Here we have triple X, where we have three X chromosomes. And Klinefelter syndrome is XXY. So this is a female. This is a female. And this would be a male. And there's a range of effects. You can have very mild, where you have a normal life and it's not as noticeable, to where you have severe physical changes and mental changes, and very often infertility. I suppose out of between single X and triple X, between Turner syndrome, if you have a single X, you have Turner syndrome. If you're XXY, you have Klinefelters, and if you're triple X, they just call it triple X. This probably has the most damaging effects, believe it or not. These two, this can be relatively mild, but all of this change is due to the result of an error in. So this was in meiosis. We have a problem here in meiosis. The other thing that can happen is the genetic code can change at the nucleotide level. So the other thing we want to talk about are mutations. So here we're talking about mutations. We talked about the fact that we have this genetic code, and we have adenine, we've got thymine, we've got cytosine, we've got guanine, and they go in any order. Actually, we're going to see that this is going to be an RNA, and in the next chapter, we're going to see it's uracil, but BBGCAA. So each of these is part of the genetic code. And as long as this code stays the same, we don't have too much of an issue. And one of the things we'll learn in the next chapter is that when we make a protein, so this is the code to build a protein. When we create this protein, we're going to read the genetic code in groups of three. It's called a codon. And to kind of represent what can happen when we change the genetic code, I have a sentence: the cat ate the rat. These are groups of 3 letters. So this represents a sentence that we might use to build a protein, although obviously we have different letters, you know, ATC, GGC. We can change these by substituting a nucleotide, deleting a nucleotide, adding a nucleotide, or duplicating a nucleotide. So let's say that I substitute an H for the letter R. My sentence then becomes: tree cat ate the rat. Now, that's not necessarily a great sentence, but I can still kind of understand what's going on. What we'll see, though, is as we do some more of these possible mutations, it gets to become more severe. So let's say we have a deletion mutation where we lose a nucleotide. So let's say in this case, I lose the letter H. I'm still going to read this in groups of 3. So I'm going to have TEC, ATA, TET, HERAT. I now have a nonsensical sentence. And so this is a pretty severe mutation because it causes everything to shift. It's called a frame shift. And we get something that is, oh, this should be nucleotide, not nucleotides. We get something that's nonsensical. And we get the same thing if we do an addition. So let's say we have an addition. Let's say I add an H. And remember, we're still going to go in groups of 3. So if I add an H, I'm going to have THH, ECA, TAT, ETH, ERAT. Off screen a little bit. So this is also a severe mutation because it gives us something that's completely nonsensical. And it's going to build a protein that is going to be completely unusable. Duplication, maybe not quite as bad. So with duplication, we're going to duplicate one nucleotide or a group of nucleotides. So if we duplicate a group, we might have the cat ate the. Still partially a sentence, but it's somewhat usable. But what if I just delete one nucleotide? Well, now I'm back into the severe mutation category. THH, ECA, TAT, ETH, ERAT. So it kind of depends for duplication if we're duplicating a single or a triple. But this really kind of gives you a little bit of a handle on how just changing one nucleotide, either through substitution, deletion, addition, or we could do duplication, can affect the genetic code. Remember, what we're going to see is we're going to read this genetic code the same way we would read the sentence. So what are our takeaways here? Our takeaways are we can have random changes in the genetic code, either at the chromosomal level by having a problem during meiosis where we get a non-disjunction, or when we are, what we're going to do is we're going to copy the genetic code from the DNA into the nucleus where we're going to form proteins. And during that copying process, we can end up with substitution, deletion, addition, duplication, and it causes a change. Now, keep in mind, not all mutations are bad. Sometimes a mutation actually builds a protein that helps the organism and is better for the organism. And we're going to talk more about that also in the next chapter. Okay, thanks for watching.